Okay, so I just realized that I said I was going to go over secondary active transport today with you guys, but I forgot that I was taking a field trip with my forensics class to the morgue. So I am going to record this little mini lecture pretty quickly. I do apologize. I have some kind of unruly kids in the background who are very close to their bedtime, but I want to get this recorded and uploaded for you guys. I guess since I'm here, it won't hurt to just review what we talked about yesterday, that primary active transport is active transport, so it's going to require energy. And primary means that that energy source is going to be come from ATP, and that ATP is going to drive a integral membrane, transmembrane pump um, in the membrane. That's what transmembrane integral membrane means. And the one that we focused on was the sodium potassium pump. So remember that it requires ATP. Of course, it requires sodium and potassium in a ratio of 3 sodium out for every 2 potassium in at the expense of 1 molecule of ATP. And it does, of course, require that transmembrane protein in the membrane, and that it's very important for, especially for your brain cells, but it's in all sorts of other cells in our body. For example, our muscles, including our heart muscle, and other cells in the body. But probably the most important is the most reliant on it is the brain cells. And of course, we looked at this. So again, 3 sodium out, 2 potassium in at the expense of 1 molecule of ATP. Again, the key part for this is that it's going against the concentration gradient. So you see sodium's highest out here and lowest here, so low to high. For all active transport, it's going to require, it's going to go from low to high against the concentration gradient. So I used the analogy in class yesterday. It's like you were, everybody got let out of a concert and you realized that you dropped your phone and you're going to try to go find it. So it's going to require some going against that gradient or going against the grain, going against the traffic, some extra energy to do that, to drive through that. All right, so again, what I was going to be focusing on today is our second part, which is secondary active transport. I don't care about any Cyber Monday deals. And for secondary active transport, remember I gave you the analogy of there's different energy currencies, there's different energy mechanisms. I'm not going to go through any specifics there, but like for my example, I said I prefer to use my credit card because I get points for that. And so that would be like how primary active transport, the primary um, active transport requires ATP, but secondary active transport uses energy sources other than ATP. Just like I could use other currency sources buy to buy things like checks or cash or debit card. Okay, so the requirements for secondary active transport are that it requires energy other than ATP, and of course it does require energy, and it's also going to be taking things from low to high. And this, if you click on your document that's on Google Classroom, you can get to this animation that I'm going to show, and it'll go through these 4 types. And these pictures here are pretty much very similar to what is going to be going on in your video. So I'm going to probably play this little animation a little bit and then stop it and talk about it a little bit. Okay. For secondary active transport mechanisms, the ones we're going to focus on are going to bring large amounts of materials into or out of the cell. So the word for into the cell, a process that brings things into the cell, is endocytosis. Endo in cytosis, in the cell. And exocytosis is going to be particles that are taken out of the cell. Exo, out of the cell. For exocytosis, that's easy. There's just going to be one process that's just labeled exocytosis. But for endocytosis, it's divided up into three different types of endocytosis. And then I'll play those here.
The substances taken in by single-celled organisms are often particles or large polar molecules that cannot cross the hydrophobic plasma membrane. Many single-celled eukaryotes employ endocytosis to ingest such food particles. In this process, the plasma membrane extends outward and surrounds the food particle. Cells use three major types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterium or a fragment of organic matter, the process is called phagocytosis.
Okay, so that process, and I'm just going to kind of rewind it a little bit here. The process of bringing in large particles like bacteria or large food particles is called phagocytosis. And so this structure here that's going to be coming into the cell is actually called a phagosome. And that is equivalent to the cell eating. So it's like taking in a big chunk of something. So the word phage means to eat. So phagocytosis is kind of equivalent to cell eating. Something large particle matter that's coming into the cell. And then it'll get digested. Okay, so let me just let him play that out.
Matter. The process is called phagocytosis. If the material the cell takes in is liquid, it is called pinocytosis.
Okay, so you might think of that liquid. Okay, is that just water? Well, it could be, but it's usually things that are dissolved in liquid, like salts and things like sugars that are from the environment that the cell is able to sense and bring in. But pinocytosis is equivalent to the cell kind of drinking. So phagocytosis is eating, pinocytosis is drinking, bringing in material that's dissolved in liquid.
Cell takes in is liquid. It is called pinocytosis. Specific molecules, such as low-density lipoproteins, LDL, are often transported into eukaryotic cells through receptor-mediated endocytosis.
All right, that's receptor-mediated endocytosis is a big, long set of words that mean that there's a specific molecule, and it shows you better on the picture that's actually on your document. But this is one-way cholesterol, so it calls this LDL. LDL is the protein that carries cholesterol. So we'll just simplify it and say this is how cholesterol moves around in the cell, because cholesterol is made up of lipid, so it's not able to just kind of move through the cell freely. It has to be brought into these large particles. And I like this picture. It shows that it's specific. So these are the receptors, and they're going to bring in cholesterol. So we'll watch that, and then we'll talk about what happens when you get mutations in these receptors. Remember, your cell needs cholesterol for a lot of different things, including the membrane stability. So to make sure the membrane's not too fluid or too brittle.
Molecules to be transported first bind to specific receptors on the plasma membrane. The interior portion of the receptor protein is embedded in the membrane. The protein clathrin coats the inside of the membrane in the area of the pit. When an appropriate collection of molecules gathers in the coated pit, the pit deepens and seals off to form a coated vesicle, which carries the molecules into the cell.
Okay, so that's receptor-mediated endocytosis. We see those little wrenches there. Those are the receptors. They're specific to this molecule, which we could say is cholesterol. There's other things that are brought in this way. This is just one example. But they're going to fill up this pit, and then when enough of them are there, it folds upon itself, and then it's going to travel through the cell that way. Receptor-mediated endo or into the cell at receptor-mediated endocytosis.
Gathers in the coated pit. The pit deepens and seals off to form a coated vesicle, which carries the molecules into the cell.
Okay, so those are the three forms of endocytosis. We have, just to reiterate quickly, we have phagocytosis is the kind of eating or ingesting of large particles. Pinocytosis is drinking or bringing in dissolved materials into that are dissolved in liquids. And so that's equivalent to like the cell drinking. And then receptor mediated endocytosis, which is specifically how cholesterol moves in the cell, is going to move bind with these specific receptors and be brought into the cell that way. And then the last part is just going to talk about the exocytosis part of it. It's going to be pretty much the reverse, and we kind of just have a generic. If it's going out of the cell, we're going to call it exocytosis. Say it's generic, but it's actually very important. This is how your neurotransmitters, these chemicals that keep signals moving in your brain, go from one brain cell to another. They leave the cell by exocytosis, and then they're picked up by a receptor later.
Exocytosis is the reverse of endocytosis. This process results in the discharge of material from vesicles at the cell surface to the outside of the cell.
Okay, so that's that. Let me pause this for a second, see if I can find another animation that I want to show really quick. Okay, so I found something that does a nice job of showing what I was talking about here with exocytosis. So in one neuron, the electrical impulses stimulate these vesicles to release their neurotransmitters into this space, and then they're picked up by receptors on the adjacent receiving cell, and that's how the nerve impulses travel from one neuron to another so fast. So you don't have this one, but I'll link it in the in your notes part on Google Classroom, because I think it does a nice job of showing nerve impulses cause the exocytosis to transmit the neurotransmitter to the adjacent cell, and then that's how the nerve impulse moves from cell to cell to cell. Okay, so that pretty much is it for the active transport. This is just a very similar look that's already labeled for you. You're going to need to make sure that you know that this is requiring energy, but not necessarily ATP specific. You can use other energy sources. And then the moving of solid particles or eating of the large particles is called phagocytosis, kind of like drinking, bringing in intercellular fluid. So sometimes you see that these things are dissolved here and it'll bring materials in. That's pinocytosis. And then what I like about this one is it shows that the receptor mediated endocytosis is specific to this star, where the star could be cholesterol or a particular protein or a neurotransmitter, whatever it happens to be. But notice that this little wrench, this receptor, is not going to be attached to the square. It's going to be specific to that. And again, remember that's how cholesterol is brought in. So phagocytosis, eating large solid particles. Pinocytosis, drinking fluid, stuff that's dissolved in fluid. And then receptor-mediated endocytosis is for things like cholesterol that are brought in through a specific receptor. And then this is just showing the exocytosis. So that the material is going to be in a vesicle. So it's a membrane-bound vesicle. You see the phospholipid bilayer here. The vesicle, it's going to merge with the membrane. So the membrane is actually going to get larger. In the case of the endocytosis, the membrane actually gets smaller because that membrane comes from the cell membrane, where in this case that vesicle is going to come from probably like the Golgi and then be transported, and then that's going to merge, and so that membrane space is actually going to get larger. But that's just a kind of an aside about that. Okay, so that's that. You should make sure, of course, that you've answered the questions with this along the way, and then for the rest of the time, I want you to work on your Edulastic. You have pretty much everything that you need for that now, and that's going to be what you're going to want to concentrate for Friday's quiz. Okay, have a good day.